and this is where we're at. And I was meeting with a student, scheduled meeting with a student just before class, and so I didn't get to do my usual last minute prep. And so one thing that's not on here is the reality that we have an exam in 2 weeks. It's our second exam, and I know what it'll cover, and I just didn't get a chance to add that. It'll cover chapters 18, 19, and 20. And we can get through those. We're nearly through 18. We'll get through that today. And then 19 is a pretty straightforward chapter for us once we navigate, through it in class. Like what do we need to get out of this? And then 20. And 20, I think the chapter is called Cancer, and it's got, that's where the book talks about cetuximab and CAR T-cell therapy, and it talks about other kind of viruses that can help cause cancer, like can lead to cancer, and it explains the molecular pathways that are involved, and Jude and Foss show up there like that. So 20 is a really satisfying chapter because we've been just pointing at it since day one, okay? Then, I did get that up there, but that's where we're heading. 18, 19, 20, an exam in 2 weeks. It'll be the same format, same hard questions like that. But I'll try to organize it so that you feel like you're not running out of time like that. We need to stay focused and use up the time, but I don't want to have people running out of time. Okay, so we heard from Riley, and we stopped, right? And I said, Tuesday ended up being kind of a review session because we said, okay, well, what are the cellular responses that we've learned about that happen in the T cell during CAR T cell therapy? And Riley talked about it. And it's important as a studying experience to take what we talk about in class and build the more directed information that would be like the answers for questions on a test, right? Like, what are the cellular responses that we learned about in CAR T-cell therapy? And we made a list of it, right? Our list ended up being sloppy, right? Because I have sloppy handwriting, but also we're talking through it, right? But that's part of studying for biology courses, is to organize this stuff. And so I think that the format for the exams in this course is a good format because it points toward that type of organizational. Take the knowledge and organize it into functional pieces of little bricks of information like that, like sort of fancy flashcards. And so that's good. Then because we talked about that, I said, hey, here's another thing. What are the targets of the CDKs that get activated at start that we learned about, right? We made a little list of that. And we had talked about those separately, so that's another good example. Where we talked about them clearly, we talked about MCM a while ago. We talked about retinoblastoma and just how satisfying that story is, like that. There's lots of people that would just be like, I encourage you to write the exam only about retinoblastoma. And you guys would just be like, that's outrageous. I had a grad school level physical chemistry final exam like that one time, where it was me and other students that were PhD students in physical chemistry. And I don't know if you've heard about physical chemistry, but that's just like a nightmare. It's just a pitiful nightmare. And the professor hands out the final exam, and he didn't say anything about the fact that it was one question worth 100 points. And it was just like that. 2 of these students, grad students in physical chemistry, got up and crumpled the exam and just threw it at him and walked out. One of them swore at the professor like that. I was there, just like, I used lightning bolts on that exam. I know this and I know this, and I'm just going to put a lightning bolt in here because I don't know how they get connected. It was just out of control. Yeah. So I'm not going to do that because I experienced that. So we're not going to just talk about retinoblastoma, but retinoblastoma, that's a target, and it's a co-repressor, transcription factor, and then the targets, the response genes to E2F are just such satisfying stories. Oh, we got to make more DNA polymerase because we're going into S, right? And so when the light turns green, we can leave that analogy or metaphor behind because it's just biochemistry. Literally, when the light is green, the action of CDK is to do these things that you are in S because you have lots of DNA polymerase and you have lots of MCM, and the MCM is getting phosphorylated and directing the DNA polymerase to the origins of replication along the DNA chromosome. And it's going to have plenty of substrate. It's going to have plenty of DNA to copy, and it's going to have plenty of the AGC2 nucleotides, like that. So reorganizing your notes and your learning is a really big deal. I record our lectures because it's painful if you miss one, because you want to get that opportunity to learn about it. Okay, so that's where we're at. Okay, so, and that's the flow. So these 3 chapters for our exam, it'll be good. And then we're also studying for the final. I think for the final, we'll be able to add just one more chapter, right? Because we only have like 1 or 2 weeks of class, so maybe 1 or 2 more chapters, and they'll be back in the book like that. More general things, like one of the chapters I'm sure would be the nucleus, and I think that's chapter 12, like that. Yep? Yes? Wait, yep?
From the first exam, everything like that as well, so like 1, 4, 8, and 9?
On the final, the second exam will focus on new material, but we've seen that new material really builds on the first. So I won't ask specific questions that you're like, oh wait, that's from back in the first part. But it's based on that knowledge, right? So, you really have to understand that play model video, because we're talking about retinoblastoma. It's a transcription factor, so you have to understand that. The final covers everything. So there'll be some of the new material in the final, but there won't be very much material, and it will be about everything. So I mentioned before that when you study for the second exam, well, we really want to know that material well, right? And one way of appreciating that is because we're going to have 2 tests in a row, and so there'll be, you'll have to know a lot of stuff about these chapters. But remember, this is the 4th section of our book, and the book is pulling it together like that, right? So everything in our class has been carefully orchestrated to try to get us to really be able to appreciate these chapters, these details, okay? So that's where we're at. And so, we're just about done with chapter 18. We're going to talk about the phases of mitosis a little bit today, and then we're going to roll on to chapter 19. But let me check in and stop and just see about questions that people have about stuff. Yes, Tiva. Sure. Yeah, a few. Yep. And because we need to be reading the book and reading those and working on those. And they will be just a few. So, the big effort is to really understand what happened in class. That requires you to use the book, right, to help you understand it. So that's the first part, is really understand what's happening in class. Even if it's mentioned once, it's, very fair game. And it could be answering a question, right, like that, like that. So everything that comes up in class, and to understand that you need to read the book, like that. And then there'll be some questions that they're really important in the book, but we just didn't get around to talking about them. So identify those next important things like that. Yep. And you can bring your book in and ask questions from the book like that, and clarify, like, what do we need to know about this section, right? Because it is going to always be 2-point questions. They can have some detail to them, but they're not going to be a question about a footnote or a trivial detail. Godwin has a question, but Brooklyn, do you still have a question?
No, I'm good.
Godwin has a question.
For chapter 19, right, do you want us to focus mostly on apoptosis because they have cloning in there, they have trans differentiation in there?
Yeah. So we'll go through chapter 19. There's a way to navigate through it to get the core pieces. It's about programmed cell death, and then it's about stem cells, like that. And other questions people have about stuff? Yep.
Are there any phases in particular from chapter 18 that you want us to focus in on, since there's a lot in the chapter?
Maybe this chapter, these 3 things. We have these 3 topics. Our super favorite is start, and we had to go through that twice, right? Because we talked going round about it, because we were bridging chapter 17 to chapter 18. So much of stuff we learned in chapter 17 was about growth and regulation of the cell cycle, that was going to be the cellular response. Under the wound, it's the chapter 17 story of Ras, Raf, Mech, Elk. And then chapter 18 is Elk is a transcription factor that binds to target chains that include binding to the regulatory region of cyclins. And cyclins are often the rate limiting step the action at start is CDK. CDK stands for cyclin dependent kinase, like that. So that was the first round. He's talking about that and talking about start. And then we went down and talked, and we moved to the other point in the cell cycle to learn just the nuts and bolts of that DNA damage checkpoint. Then we went back to start, and we learned some about the small inhibitory proteins, and we learned about some tumor suppressors like retinoblastoma, and we learned about some more about the targets of the action that happens when CDK is activated. So that's what we're talking about. And you said, do we want to know some things about the phases? And so are you referring to this part?
I guess I meant like from like S.
Yeah,
G1,
that's a
S,
good question.
S.
So we talked briefly in class about what happens in G1 and S and G2 and M, right? Like that. And so just to talk about that, S is we got to copy the DNA, and it takes a while. Typically, in one of our cell body cells, it might take 6 hours to get through S, because it's just so much DNA that needs to get replicated. Okay? And the cell is really focused on that. Cells don't hang out in S ever. They go through it. It's a task, and they're completing the task of replicating the DNA. They start S with the cells being diploid in our body, and they end it with the cells being tetraploid. They start it with the homologous chromosomes, one from mom, one from dad. They end it with homologous chromosomes that have been duplicated, photocopied, and made into sister chromatids that are going to separate at M, like that. So that's S, and it's all about that. And you know, what do we need to get that done? You know, the list of the biochemistry of getting the job done and replicating the DNA. Then I said G2. G2 in vertebrates is really typically just a phase they're just moving through it. They don't really wait in it. There's no such thing as a G0 in G2 for vertebrates. In invertebrates, G2 often is a place where they wait. They wait for a hormone signal. There's a control point, you know, an external control point in G2 in some invertebrates like that, there, you know, at that point. So then M phase is also, like S, a thing that is just getting through it, just going to get through it. There's no real gene expression, no real living activity that happens in M phase. They're just getting through it. We'll talk about the steps now, you know, like that. And so those are those three phases, and we want to be able to list, you know, what happens during them, right? And you know, and for those, it's pretty straightforward what we learned about, right? G2 is, well, there's the G2 to M transition and the DNA damage checkpoint. So it's like that, right? So what happens during those phases? Then G1. G1 is where the cell really does its living. When we think about cells and their living, they're in G1, and they're there for variable lengths of time, so much so that they talk about G0, right? Our brain cells are in G1 and G0 because they're just going to be parked there forever. Our other cells, like our stem cells, our muscle stem cells, they're in G1. They're waiting for, you know, some damage or growth or, you know, activity to be stimulated. So they're waiting. They're in G1 and G0. Our muscles themselves are differentiated. They're going to be in G0 doing the muscle contractions for our life. So lots of stuff happening in G0, all the different transcription, gene regulation, cells migrating and doing stuff is G1. Yeah. Then G1 also is the home for the G1 to S transition start. So in a list of the phases, you know, we just go boom, 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 boom. Yep.
I just had a quick question. You said with G2 cells that they can wait for like a hormone signal before they like leave G2. We talked about it earlier or a couple weeks ago how some cells like wait until you hit puberty to become active and go through the cell cycle. Are those cells that are waiting until you hit puberty in G1 or are they in G2?
So yeah, so what I said is that in vertebrates, right, us and mammals and all the vertebrates, all the control of hormones is in G1 at start.
Okay.
Yeah. In vertebrates, there's just tremendous diversity of life in invertebrates. And in some invertebrates, there's more stories in G2, like including even a hormonal control of the cell cycle in G2. But in vertebrates, in invertebrates, right, vertebrates are all very similar like us, and the big show is right at start, and it's that CDK regulation. Our brain cells are in G0, but they're really parked at the red light. You know, they're just waiting at the red light permanently. Yep.
Are those that he talked about not at G0 because they're at start, or?
Yeah, so people talk about G0 because it's fascinating to see a cell permanently stopped at a red light, like a brain cell or any differentiated cell. It's also fascinating to see the cells in our childhood that are waiting for puberty, just waiting for estrogen or testosterone. They're waiting at a red light that's going to become green. The hormone's going to turn to green. So they'll sit there for so long that scientists just go, you know, it's like a detour. It's like a G0 is a detour, you know, a special path. It's not. They're just waiting. But it's just hard for even, you know, advanced scientists to go, really? Just waiting at a red light when they do their whole living? And the answer is yeah, that with regard to the cell cycle, they're just parked. They can do all kinds of stuff. Cells can crawl and contract and die and so forth. You know, so that's why G0 comes up, is because it's just a thing where you're like, come on, it really seems like a different thing. Yeah. So that's that. And within those phases, right, we say, oh, brief talk of S, brief talk of G2, somewhat brief talk of M, and then more talk of G1, and especially in the chapter 18 that focuses on that red light. That red light is the regulation of CDK. When the light is red, those CDKs are inactive, and then they get activated, and they immediately do their job. They're kinases with targets that are going to phosphorylate, and that's what it means, literally, biochemically, for the light to be green, is the CDKs are active, right? And we saw that the CDKs start and the light turns green kind of like over a little bit of time, because they tend to go through a positive loop. And some CDKs get activated and they activate more. They phosphorylate and target B27 for degradation. There's lots of stories like that. So the light turns green, and it's all about CDKs, and CDKs doing their action, and they actually, the light starts to flicker green, and then they're just like, we're turning this thing green and making this happen, right? Okay? So in mitosis, we just want to talk, because our book talks a lot about this, and it's a story in cellular biology. Scientists have been able to look in a microscope and see events in M phase for 150 years. They've seen the chromosomes condense, line up along the metaphase plate, and separate during mitosis. And so our book spends a lot of time on this because it's been studied a long time. It's really good cell biology, right, like that. And we want to breeze through it. So we're going to breeze through it in class, but this would be a place where there would be probably a couple of questions from the textbook that we just didn't get around to talking about in class, because the book talks about it a lot, and it's good, understandable cell biology. Right? The book talks a lot about the mitotic spindle. It's the microtubules that grab the chromosomes and pull them apart. And there's a lot of talk about the mitotic spindle because it's the microtubule elements of the cells, the cytoskeleton, and at the base of the mitotic spindle are the centrioles, which are fascinating microtubule organizers. So we're going to breeze through this, but also say, but you got to read it, try to get more out of it, ask questions about it. So these 4 stages, right? Prophase, metaphase, anaphase, telophase. And mnemonic is IPMAT. So IP is for interphase, because we're looking under a microscope and the cell is in interphase and spends most of its life in interphase, because interphase is G1, S, G2, like that. So interphase, IPMAT. Interphase, prophase, metaphase, anaphase, telophase. And for sure, we'd like to be able to say one thing that happens during these phases, and maybe a little bit more, and we're just going to go like this. So prophase is the start of M phase, and the most famous thing is that the chromosomes condense. And we love that because when CDK1 at the G2 to M transition, gets activated by removal of inhibitory phosphates. It's a kinase, and it starts phosphorylating its targets. And some of its targets are histones. It phosphorylates histones, and they fold closer together, and the chromosomes condense. So seeing the chromosomes condense to us means CDK1 is active, because that's literally the action, right? Another thing in a lot of cells, including human cells, is the nucleus breaks down. And we saw that's because of CDK1 action. So the start of mitosis is prophase, and it's because CDK1 gets activated. And what we see occurring is the result of direct action of CDK1 phosphorylating targets. So that's prophase, like that. And the famous things for us are the nucleus breaks down, the chromosomes condense and become visible, and we know why. And that's just a really important take-home message, because all of this is just straight out molecular biochemistry. And you can see this stuff happening under the microscope, but it's because of molecular events that are below the visible range, like that. So then, that's prophase, like that. Then metaphase, chromosomes line up. So they line up along the center of the cell, and along a place called, there's this, a line called the metaphase plate. And so, that's another very visible thing. The chromosomes get wiggled around, and then we see them, and it looks like this, where here are the chromosomes, and they're all lined up. And that's called the metaphase plate. And so that's the famous event that happens in metaphase, is we get the chromosomes lined up along the metaphase plate. And we can draw in here, they're lined up because they're connected to the mitotic spindle, which is composed of microtubules, which are like strings, like tethers that are connected to the chromosomes and helping organize them. And the microtubules are organized that way because at the base of them are the cell's centrioles. So for metaphase, we'd want to describe that and maybe draw a picture of it, write a little bit about it, like that. The book talks about it a lot because it's fascinating to see it. Okay? Then the next thing is anaphase. And in anaphase, the chromosomes separate. And they're separating because the microtubules are pulling them apart. So they're getting pulled to the opposite poles of the cell. And then the last one is telophase.
What did you say pulls them apart, the microtubules?
Yep. And what people do when they talk about telophase is they always talk about this. They always say, well, it's telophase and cytokinesis. So let's start out with telophase. So telophase is going to be the ending of M phase. And we know what that means. M phase is going to end because the cyclins get degraded. The CDK has been active all throughout M phase. And to leave M phase, we need to inactivate it. And the way we inactivate it is we degrade the cyclins. CDK stands for cyclin-dependent kinase. We stop its activity, in this case, by degrading the cyclins. So that's a famous thing in telophase. The biochemistry is the cyclins get degraded and inactivate CDK1. CDK1 is present all the time in every one of our cells, and only active in the M phase, and it's because of the removal of inhibitory phosphates at the start and the degradation of cyclins at the end. Okay? So we leave M phase, and as a result, telophase must involve reforming the nuclear membrane, the nucleus, and decondensing the chromosomes. Okay? that's telophase. That's what we're going to get out of telophase. That's part of M phase. Then, happening right in an overlapping way with telophase, so they say telophase and cytokinesis because these overlap in time. And we just learned about telophase, and that's our natural focus, right? Good thing. It's the ending of M phase. So the inactivation of CDKs and the reversal of what it's been doing. They've been, keeping the chromosomes condensed, keeping the nucleus disrupted, and other things too, right? Like that. The book lists more actions that happen during M phase. Cytokinesis is just the fancy word for cell division. And that begins during telophase. Cytokinesis is not really part of M phase. It overlaps with it some. The cell starts to get ready to divide. But it's really a separate action at the end of M phase. Okay? And so let's just mention one thing about cytokinesis, and that will be in this slide in the book, because our book talks about all this. And that is, here's cytokinesis, and the famous thing about cytokinesis is there's going to be a pinching here to separate these two cells. You can see here that M phase is getting completed, right? We're starting to see the nuclei reforming, and we're no longer really seeing the chromosomes, and we're separating these two cells. And we had learned about this role of the microtubules in the M phase, pulling chromosomes apart. This contractile ring is made up of another cytoskeleton component, actin. And actin is part of not microtubules, but actin is part of microfilaments. So in mitosis and cytokinesis, we have these big roles for cell cytoskeleton, and we have a role for microtubules, pulling the chromosomes apart, and then a different role for actin. Actin is not microtubules, it's microfilaments. And it's hard for students to distinguish those. I like to try to remember and always say to myself, when I say microfilaments, I always say actin microfilaments, because that just helps me remember. And what they're like is, it's like the actin and myosin that's involved in muscle contraction. And the actin are long polymers. So we get long polymers forming here. And then the myosin proteins slide them past each other. And so just like muscle contraction, where our muscles shorten, these actin filaments slide past each other, and we get a contraction. It's called the contractile ring. We get a pinching off of that. Those are inside of the cell, and they're organized in a little loop, like a little lasso that closes and pinches them together by that contraction, which is comparable to our muscle contraction. Okay? So that's a nice thing that people like to test about, is the role of the cytoskeleton in M phase. Oh, it's about the microtubules. And the role of the cytoskeleton in the cytokinesis. Oh, it's about actin microfilaments. Like that. And our book lays this out really well, like that. And it's important, like that. If you look at the chunk of the chapter the book uses to talk about this, it's important stuff. But so we'll have questions about it, and more than we talk about in class. But I just want to, I think it's not as hard to learn this as it was to learn about starch and G1 to S transition, and also the G2 to M transition. Okay, because with that, I'm really kind of done talking about this. The book makes this comparison between meiosis and mitosis, and let me just show you that in a way that is satisfying to learn. So what we've learned about in chapter 18 is about what's called the mitotic cell cycle. And in eukaryotes, there are two mitotic cell cycle. That's what we've been talking about all this time. There are two types of cell cycles in all plants and animals, and in all eukaryotes, there's the mitotic cell cycle, and that's what builds the bodies of animals and plants, and that's what causes yeast to, increase in number. And then there is meiosis and the meiotic cell cycle. And meiosis is when gonads form, not gonads, gametes form, and we're used to calling those sperm and egg. The general term for them is gametes. And in meiosis, what we're going to do is we're going to produce cells, the sperm and egg, that are haploid. Up here, the mitotic cell cycle, they're really typically starting with diploid cells, and then we get tetraploid, and then we get back down to diploid. And there's also that N letter nomenclature. So these are called, diploid is called 2N, tetraploid is called 4N, and then back down to diploid 2N. And the N is really indicating like a genome. So haploid cells are 1N. And we're going to get the sperm to fertilize the egg and go from 1N gametes to 2N fertilized egg. And so the book spends a little bit of time talking about meiosis and the cell cycle that involves creating the haploid gametes, sperm and egg. And so we should learn about that, and the book doesn't show it this way, and that's too bad because this is a really satisfying thing to see. And it goes like this. We're used to drawing the cell cycle as a circle, like that, but it's really a timeline, right, where start is here, and then we have S, G2, M, and then S again, or G1 again, like that. So although it's drawn as a circle, it's really a timeline. And you could draw it as a timeline. Well, let's just draw it like this. So we'll draw it as a timeline like this. Or we say, okay, so let's draw a timeline. And this is mitosis and the mitotic cell cycle that we've learned about, right? So you'd have, here's a cell cycle, and it goes from, start. And then that means we'd have S, G2, M, G1, and then we'd be back at start. And we'd go S, G2, M, G1. And then we'd be back at start. And we'd go S, G2, M, G1. That's what we've been learning about the last couple weeks. So it's nice to draw it out that way, because it becomes very easy to compare and contrast mitosis with meiosis. So here's meiosis, and it goes like this. There's a start, and we go through S, G2, M, G1. Like that. And this M phase is called meiosis 1. And it's a little bit different than mitosis. And the difference is kind of like a bookkeeping detail that we don't need to dwell on, we just need to be able to say. During meiosis 1, the sister chromatids stay together. Those are the photocopies, and in mitosis, it makes a lot of sense to separate the photocopies. Each of the new cells should get, the photocopies of each of the chromosomes. But the sister chromatids stay together here. Meiosis one, there's one other thing that would be mentioned here. Meiosis one is also where something called recombination or crossing over occurs. So as we label it, the meiotic cell cycle looks really similar to mitosis. You know, we start, we go S, G2, M, G1. But that M phase is different. The sister chromatids stay together, and during meiosis one, the chromosomes undergo this recombination. Then we have the next cell cycle, and it goes like this. Here's start. Then, a kind of a key feature, and there are just a couple of key features, is that there is no S phase here. We skip S phase and we go right to G2, M, G1. This M phase is called meiosis 2. And in this one, the sister chromatids now separate. And that's it. So the real key things boil down to this. Between meiosis 1 and meiosis 2, there's no S phase. So that's how we're going to drop down in the number of the chromosomes, is we just don't replicate them. You know, so we go through, but we don't drop them down, right? We went from diploid to tetraploid, and in meiosis 1, we dropped back down to diploid. So now we're diploid, and in the next round, we just don't copy them again. So meiosis 2, we go from diploid down to haploid. Right?
Does
So
recombination occur in the second one?
what?
Does recombination occur in the second one?
No. Yep. And so when you talk about meiosis, there are really these two features. One is we're missing an S phase, and the other is that in meiosis 1, the sister chromosomes don't separate. Okay, that's not a big deal. But recombination occurs in meiosis 1. So those are really the two features of meiosis that most dramatically distinguish it from mitosis. Yep?
Do you mind repeating, did you say it starts as haploid or starts?
Yeah, let's just draw in those numbers, that haploid diploid number thing, and what we'll do is instead of saying haploid diploid, we'll do that 2N thing. So up here in S phase here with mitosis, we would have started as 2N and then gone up to 4N. Diploid up to tetraploid, and then mitosis occurs, we go back to 2N. Then another S phase, so we go up to 4N. Then in mitosis, we go back to 2N. Then in S phase, we go up to 4N. Then on mitosis, we go back to 2N. So that's the cell cycle. We go from diploid to tetraploid to diploid to tetraploid to diploid to tetraploid. Okay? So then,
Start is 2N and then at M phase or before M phase is 4N?
yes, yeah,
And
right. Mitosis is when we're going to separate the chromosomes and make two cells. So we got to get up to tetraploid and drop down to diploid when we separate the two cells. Then, corresponding down here, we got this. So in meiosis, this is going to look really similar. 2N up to 4N. Then this meiosis 1, we drop it down, but you'd put 2N in quotes here because the sister chromatids are together. So it's a different situation somewhat. And we're at diploid, and then here we skip S. So when we go into meiosis 2, we're at this kind of so-called 2N. We have that many chromosomes, but they're actually mostly, half of them are, top duplicates. And then we go through meiosis 2, and that's how we drop down to 1N, or haploid. So diploid, tetraploid, diploid, tetraploid, diploid, tetraploid up there, then down to meiosis 1, diploid, tetraploid, down to a kind of different diploid, and then down to haploid. Okay? So the book shows it like this. It shows it as a visual, not helpful, I don't think. I think just you want to do the counting and you want to highlight those two features. No S phase between meiosis 1 and meiosis 2, and meiosis 1, yeah, this is two chromosomes stay together, okay, it's kind of a detail, but more significantly, that's where this recombination takes place. And recombination or crossing over is a big deal in genetics, and a lot of talk about that in genetics. It's not a course in genetics, so we're going to freeze over that fact. Yep.
Is that because there's a lot of like genetic issues that happen during recombination?
So the interpretation of crossing over is that when we have long chromosomes, the chromosome from mom has all the genes on it, and the chromosome for dad has all the genes on it. But because they're physical chromosomes, each of those genes are linked physically together, so that when we mix the parental genotypes together, they really wouldn't mix completely, and we'd have a lot of mom's alleles getting together, going to one offspring. And to interpret it, natural selection, evolutionary function of recombination is we're just going to stir mom's and dad's genomes together so we get more diversity in their offspring. There's still going to be, all the kids are still going to be, half mom, half dad, but more of a stirred together thing where it's not just half of mom's chromosomes and half of dad's chromosomes, but half of mom's genes and half of dad's genes. Yep?
Does this process work the same way, like within mom and within dad to make the haploid cells?
Yes.
Okay, so
And that's a good question, right? Because one thing that's interesting is we're used to humans where we are you making sperm or are you making eggs? There are plenty of vertebrates who have very similar hormonal regulation of sexual differentiation, where an individual fish, for example, in many species, can just switch sexes depending on environmental factors. So the molecular events, like you're asking about, oh yeah, same in sperm and egg. It's really the cellular event of, are we making an egg or are we making a sperm, that are different. In us, these cells and other mammals, these cells that form the sperm and egg, those cells originally appear when we were in our mom's tummy. They originally appear out in the placenta and crawl into our bodies and into the region that's going to be the gonad, that's going to make the ovary and the testes. And the hormonal environment tells the cells they're going to be in a female with an ovary or a male with a sperm. So what you asked about, the answer is simply yes. Same story in sperm and egg. But then that actually underscores the story of the variety of events that happen in vertebrates that can vary even in a single organism's lifespan. They can just easily change sex like that. And they change sex pretty easily because, like say in those fish, they have external fertilization, so they don't have a womb and they don't have internal fertilization with like a penis. They just release gametes, they just release egg or sperm into the environment. So the fish can just change on making sperm, making eggs like that. Yep. Yeah, and it's just part of their natural evolutionary, natural selection where they form and they differentiate or can change their sex like that. So it's fascinating to see that. And of course there's some, public topics, public discussion about gender and sexual identity and stuff like that in humans. And, it's just fascinating to see, well, it's all based on these hormones and factors. And of course, those, vary. And we learned about how a person can have hormone therapy because all male humans have the estrogen receptor. It's repressing female secondary sex genes. And it's, because there's no estrogen. But the receptor is there functioning as one of these nuclear receptors, repressing it. And if we added estrogen, gave injections to that male, they start to have these secondary sex characteristics. So it's just fascinating in that sense that, you know, this biology and this wonderful diversity of life on Earth with vertebrates, you know, is reflected in our, you know, our public discussions about, you know, what does it mean, you know, to have a gender and things like that, you know. And, you know, people are like, oh, no, you know, it's just male and female, and, you know, have these certain perspectives, and you're just like, well, you know, maybe you could, you know, learn a little bit more of the diversity of life on Earth and appreciate just how rich a pageantry is out there in reality. Yeah. So anyway, just a little bit of that, little meiosis in there. Yeah. Other questions people have? Because that's the end of chapter 18. So now we're just going to go rocking into chapter 19. And chapter 19 is about programmed cell death, which gets abbreviated PCD, and about stem cell biology. And it comes up here, and I mentioned before, it comes up here because lots of times cancer cells are not simply growing out of control. The cancer cells are also failing to go through programmed cell death. All right? So that's why. Why did it fit in here? Then stem cells is really, that's a new field that is just exploding, right? And so the author, you know, in some, you know, early editions of this book, they didn't have any stem cell biology in there. So it's coming in, and they decided to put it here, you know, kind of plopped it down here like that. They'll eventually have a bigger chapter, but, you know, we deal with them together. There's a reason why they put it together, but, you know, why they put these together in the same chapter. That's kind of a detail. It's definitely a detail. So we start out with this term, PCD. So there's a good chance that you've heard the word apoptosis before. Biologists, you know, don't really like to say apoptosis if they're not sure, because there are actually two types of programmed cell death in animals, and one is called apoptosis, and the other is called autophagy. In the cancer biology topics, where cancer is growing out of control, we've talked about this. The cancerous cell is failing to understand that it's out of line and it should just kill itself. And that's what apoptosis is. So that's what only the author talks about in this chapter. There's another kind of programmed cell death that's fascinating and profoundly significant in our health called autophagy. And our book talks about it in chapter 12. You don't need to read it. It just talks about it a little bit because that's also an emerging field. But you could look at it, and autophagy is interesting because it's not just programmed cell death. It involves recycling. So let's say for a person that is malnourished, they will be recycling parts of cells to generate some new building blocks, like some amino acids, because they don't have it in their diet, like that. So it's a really natural process. Autophagy are really involved in a lot of development. There's a lot of autophagy in our bodies when we're going through like fetal development. Our bodies are really changing like that. And so autophagy is hugely significant. Other natural processes like insect metamorphosis, the caterpillar just gets destroyed and you make a butterfly, right? That's a bunch of autophagy. And autophagy is super cool like that. But it's not in this chapter. So we're going to start off, we're just talking about autophagy just briefly. And what we'll do is we'll compare it with apoptosis, and then we'll talk about apoptosis like our chapter does. I really often call it programmed cell death because a lot of people just are like, let's not really focus in on the distinction here between apoptosis and autophagy, like just call it programmed cell death. So there's a good chance you may have heard of apoptosis in some other courses, and but maybe never heard of programmed cell death or autophagy, and that's how they fit together. It's a simple situation. Here's the next thing. It's really easy to distinguish between them in a drawing. So here's the difference between apoptosis and autophagy. And this is apoptosis. And in apoptosis, the cell kills itself. So, you know, sometimes people say it's like a cell suicide, because it is. The cell is dying not because it's old or injured or malnourished, but it could be a healthy cell that just is going to kill itself, right? Like that. So the cell does, kill itself, but here's the thing that happens is that another cell will phagocytize the dying cell. So that's apoptosis, is the cell kills itself and leaves just a big mess. And then the body's like, we want to get those and recycle those, those amino acids and phospholipids and stuff. So that's apoptosis. A key hallmark feature of apoptosis is the cell is going to die, and then another cell is going to ingest it, and recycle its parts. Autophagy is like this, where you'd have to draw it sort of like this, where it's kind of like this, where you'd draw it like this, and then you'd kind of go like this, where it's the cell is phagocytizing itself. So that's why it's called autophagy, because it's self-phagocytosis. And it ranges from the cell deciding, oh, there's some malnourishment or some metamorphosis or something going on. So I'm just going to ingest some of my parts. Like autophagy could involve the cell deciding, I have several mitochondria, and I'm going to just destroy some of them. And it does that by autophagy. So autophagy ranges from mild breaking down and ingesting internal parts of itself to death, where the cell just destroys itself and consumes itself up and just starts recycling itself down to building blocks. And so that's a nice distinction about them. Who's doing the actual phagocytosis, the cleaning up? And in apoptosis, it's another cell. In autophagy, it's the dying cell that's really just cleaning itself up, destroying itself as it dies. Yep?
I've heard about autophagy being like recycling the like less quality material in itself. How does it know what's good quality and what's not good quality?
Yeah, so that would be a big detailed biochemical pathway, and I don't know good examples of that to share. Like how does it know what to target? Yeah.
Okay.
Yeah. But that is because that's actually part of the aging story, not just in humans, but in all animals and even some single-celled organisms like yeast. So the yeast that causes bread to rise actually gets old and dies like that. It's just super fascinating, and it would be at this cellular level where there are some aspects of these kind of cellular mechanisms that are occurring in all animals. Yep. Okay. All right, and so the next thing, just to show on this, and that's the distinction. The next thing is we'll look at autophagy just for a moment. Okay. All right. It's in the early part of our book, and it dealt really briefly here. And so I'll just show you this little brief thing, and it's got a lot of details, but just briefly. So autophagy. In autophagy, the way the cell consumes itself is it makes a new organelle. So it's creating a new organelle that is the thing that closes around some of the cell parts and breaks them down. And the new organelle is called an autophagosome. And all of our cells have the potential to make it, and it's not really physically present, and then it just kind of materializes like it gets decloaked and shows up by assembling these components, and then it just closes around some stuff. So that's the visual look of it, right? When you say, wow, that was a weird drawing. And what it looks like is you actually get a little Pac-Man autophagosome inside the cell. It just goes around some of the components and breaks them down. And in some cases, there's some targeting of the components like that, where you're like some damaged components type of stuff like that. Most of the time, I think it's untargeted. It just grabs part of the internal cell and wipes it out like that. Yep.
So like maybe for instance like a mitochondria is damaged and then it maybe like releases something to let.
Yep.
Okay.
Yeah. So there's the idea of targeting. Most of the time, I don't think it's targeted. It's just like we need some building blocks. I think some autophagy even happens in cells between meals. It's a weird thing because you're like, wow, it took a lot of energy to build those, organelles like mitochondria and ribosomes and stuff like that. And so we'll just break them down. And what it's trying to do is just generate building blocks. Need some new amino acids, right? Because things are changing, and so it needs some new amino acids. We're just going to reposition things. Yeah, autophagy is cool, right? And you could see there's a lot of regulation of it like that and stuff like that. But that's the thing. And you could say, what do we need to know? Well, in autophagy, a cell really chews up part of itself, maybe to the point of dying, and then it does that by creating this organelle called an autophagosome. And we're done talking about autophagy. Okay? So now we're just going to talk about apoptosis. And here's a nice image about apoptosis. And this is the dying cell. It's dying. And then here it's showing another cell phagocytizing it. So now we're going to talk about apoptosis. There's going to be two cells, a dying cell and a phagocytic cell. So I still have that drawing, right? We talked about autophagy very briefly. This thing is really an internal organelle called an autophagosome. And it could be just part of the cell or a lot of the cell. And it could just be a recycler, or it could just wipe the cell out. Yep?
And apoptosis is also recycling, right? Like,
There's always, programmed cell death is always a thing where we're going to destroy some cells, but we're not going to throw away those building blocks, right? We're going to recycle them and reuse them. Yeah, always. We're not, Godwin?
So, what is the benefit for the cell if it goes through phagocytosis or?
So for autophagy, the benefit is sometimes for the cell, and the cell just needs some new building blocks because it's changing its activity. Like the caterpillar turns into a butterfly, and sometimes that involves total death. In other cases, you're just remodeling and reorganizing. You were a really big muscle, but you're not going to be a big muscle. Like that. In other cases, you're going to completely destroy the cell. In autophagy, a lot of times the recycled parts are also shared, right, with the rest of the organism. So a person who's starving, right, and like their muscles atrophy, that's a bunch of autophagy. And the recycled components, like again, amino acids, phospholipids, sugars, they're not just used by that cell, but they're shared with the organism. Because in a starving person, they're just going to break their muscles down and get some of those nutrients up to the brain and the heart, like that. So that's the thing. Not just it's with the cell itself, but also could share it, like that. Okay. So it's a drawing. There's autophagy, then apoptosis, right? And so this is a figure from your book that shows that, and it says, well, how does this cell know it should eat that cell? And that's one of our key things, and it's shown here. And it's just a crazy fact. And that is that the dying cell tells the phagocytic cell to eat it. And the way it does that is that in all of our cells right now, we have a phospholipid bilayer, right? So this, we always draw the membrane, it's just a little line, but it's really a phospholipid bilayer here around this cell. And every one of our cells is like this right now, right? Every cell. And in a phospholipid bilayer, you have an inner and an outer phospholipid layer, right? That's hydrobio stuff, right? Phospholipid bilayer. Just to give these terms, the inner layer is called the inner leaflet, and the outer layer is called the outer leaflet. They're actually made up of a variety of different phospholipid molecules. And every one of our cells right now has in the inner leaflet some of a specific type of phospholipid. In the inner leaflet, healthy cells have a phospholipid only in the inner leaflet that's called phosphatidylserine. And we have to learn that name because this story is nuts. Because what happens is that in a membrane like this, you probably have learned that the phospholipids are, it's called a fluid mosaic membrane. It's made of phospholipids and proteins. That's the mosaic part. The fluid part is, these things are just held together by, hydrophobic, actions, hydrophilic, hydrophobic. So it's very fluid. If you marked one of these green phospholipids, it would be over on the other side of the cell in a few seconds, just randomly diffusing. So the phospholipids are moving around a lot in the inner and outer leaflet, but they never flip. We never see a phospholipid flip from the inner to the outer, or vice versa. Because to do that, the hydrophilic head would have to cross through the hydrophobic environment. So it never happens. So every one of our healthy cells right now, and every healthy cell in every animal on our planet, has phosphatidylserine in the inner leaflet. And when a cell goes through apoptosis, it flips the phosphatidylserine to the outside, to the outer leaflet. And that's the signal where the cell that eats it says, dead, dying, you're gone. Because the cell eats it even before the apoptotic cell is fully dead. And so we're going to need some help flipping it to the outer leaflet. And there's going to be a protein that does that. And you probably have never heard that idea before, but that happens in cells for a variety of purposes. And so there are a variety of proteins that can flip a phospholipid from the inner to the outer, or vice versa.
Is that serine or series?
Serine.
With an A.
Yeah. And so we're going to have a little concept chat here, but you can see that's what the book is trying to show here. There's the phosphatidylserine in the inner leaflet exclusively in the healthy cell. And then part of apoptosis dying is that it activates a protein that flips it out. And see, it shows it here too. Here's the phosphatidylserine on the inner leaflet, and it flips it out, and the cell that's going to phagocytize it has a receptor for those. And it's like, okay, you're not happy, are you? Right? You want me to eat you up. Okay? So that's one of the things we're going to learn about, apoptosis, right? The cell's going to kill itself, and part of the killing is to let another cell is eat me.
It's a hit on itself.
Yeah, the analogies and metaphors are harsh, right? Because it is a kind of really depressed cell kind of situation, So we want to be, positive and stuff. But that's what's happening here. The cell's like, I'm out of line. I'm dying. Get me out of here. Yep. Yeah. Yes, and maybe yes. And then we'll have a concept check. Ellie.
Phospho something serine, that
Yep.
guy. So are you saying that there are only like a couple of them on the inner membrane?
There are some. There are a bunch of different phospholipids, and that's one of them. So there are lots of molecules of it. It's not the major phospholipid, it's a minor one, but there are, tens of thousands, in a cell. Yep. Yeah, so I think that was there a question over there, or no? Yeah, Joe?
I just had a question with like phagocytic cells. If they're like eating a cell that's like, let's say it has like damaged DNA, like it was maybe going to become a cancer cell, and they eat it, how does like if they were consuming like damaged DNA from another cell, how does that not alter like their DNA? Like kind of like if they're eating a cell like a cancer cell, how does the cancer cell that they consume not affect them? It's like, who would eat them?
Yeah.
And like, how do you just have like this like I guess cascade of like consuming damaged cells, but you yourself never become damaged?
Yeah, so when a cell phagocytizes something, it actually fuses the phagocytic material with a lysosome and
Okay.
puts it into an acidic environment.
Okay.
And so it gets digested just like inside our tummy.
Okay.
And the answer would be that, well, we could eat, there could be something like some meat that had cancer in it or
Yeah.
DNA damage in it, how do we prevent that? And the answer is the first step is we every organism always breaks things down to single nucleotides.
Yeah.
And then through metabolic pathways, the ones that are intact can be recognized by enzymes and used by DNA polymerase.
Yeah.
And ones that aren't go through these other kind of shunt-like metabolic pathways where they're further broken down to individuals who are smaller components that can be used to resynthesize. Yeah. Let's have a little concept chat action. I forgot to hand out. How's that ticket buying going? Anybody have a ticket?
Yeah, I got you.
The diet says, there'll be guest passes.
I ordered you some.
Did you buy me one? Do you have Venmo?
Yeah.
Jeremy just have to do, and I have to do a little side transaction. All right, wow, you guys are all like, wait, can you still buy for the professor a ticket? Oh, you can only give 1 A a day. I'm only allowed to give 1 A a day.
You have some friends who are bring?
Yeah, that's right.
Can we make a class trip?
No. I didn't buy him a ticket, but I went to the show and I bought him a beer, and I'm like, okay. All right, so let's just do a concept chat.
It's just a process. I guess like,
Franklin, how's it going?
they can't think about those things, they just like, I guess something happens.
What do you say for you?
I can't do my only duty, recycle me.
It's too late.
Yeah, and how it doesn't mean that external sources has
They separate.
Also a question.
Yeah, for the basis
I thought it was cool.
definition change from contract overriding, understanding the performance, contributors, and my face
You know how you have my cast?
Oh, myosin?
Myosin isn't part of the tractile ring, is it?
Myosin is.
It is?
Yeah, actin and myosin always play together. Actin is like the long fiber, and myosin is like the molecular motor that slides actin fibers past each other.
You're like little ball bearings.
Yeah, that was the big takeaway with the difference. Meiosis too.
Wait, is that like literally because of your handwriting?
And how
It's not like, wow.
Did you draw that? Or did you
I know, I drew a little circle.
They try to know how to teach you talking about when people are like that or stuff like so
I don't get it.
It's where the records start your vital start your muscle first.
We'll just do this for another minute, and then we'll have time to roll forward a little bit.
like I wonder how long it takes fast. Like you might say that people do fast, but people fast. I wonder like how long it takes for cells to get go.
Yeah, that's
Because I'm assuming they must go like something like that. We just have like go down to like energy constant. But I wonder how long that lasts.
Yeah. Where it switches from your fat reserves with your second to the protein. Listen, that's the build. Listen, if you figure that out.
All right, so let us just talk about our stuff that we're learning about. Let me just mention today, a lot of this is cell biology that we're talking about today, and I feel that like a lot of students really like cell biology. Cell biology topics are just like, whoa, that's messed up. And not so many people like molecular biology, people like me. And, molecular biology is the bedrock, no nuts and bolts of it all. Like, right? Because we're going to talk about, well, who does the flipping there? Who flips that phosphatidylserine, right? So we're going to drop it down. But anyway, cell biology rocks, right? So another possible tattoo idea, class tattoo, would just be cell biology, the big old starburster, like an American flag with cell biology on it, right? So let's talk, not to this group of 4. What's wrong with that group? Right? Let's talk to.
We're tetraploid, man.
We're in G2. Yes. Oh, sure you did. Eva,
Yes.
what's something that you and Tiva, 2 people talked about?
We talked about the importance of the difference between mitosis and meiosis and the different cell cycles and how in mitosis or meiosis it starts out.
Yep. Yeah, we mapped those out. Meiosis versus mitosis, and they boil down to be really similar cell cycle stories. A lot of the same cycle regulators, like the CDKs, are involved in both. There's a couple of features that distinguish meiosis from mitosis. So that's awesome. Amal, what is something that you and Godwin talked about? I have to jump up. And it's not your fault. I just have attended concerts and listened to too many loud tractors and stuff. And also too old. Apoptosis are all about phosphatidylserine. That's one of the hallmark features of it. Every cell's got it on the inner layer. Apoptotic cells have it. With that. Yeah. And Max, what's something that you and Joe talked about?
We talked about a couple of things, but to start, we talked about the definitions of like phases of mitosis, the mitotic plate, microtubules, microfilaments, and contractile, right? Focus more on definitions for that side of things.
Yeah.
In the stages, and then when we talked about apoptosis and autophagy, we definitely highlighted the fact that you needed an external cell to perform apoptosis.
Yeah.
So it was interesting needing something like an external force for a mechanism to go through.
Yeah. So it's a couple. You're right. Those are different, right? We went from chapter 18 to chapter 19, but different. Definitely big things we talked about. So we do have a couple more minutes, 2 minutes. Let me just show you this. Next slide. So the book shows this. These are some of the events that occur during apoptosis. And we will learn 3 of them. And one of them is this one. And the protein that flips phosphatidylserine is called scramblase. Because we go from biochemists naming things by numbers and letters to cell biologists who are like, let's call it scramblase. Scramblase is in a family of proteins, paralogs, that are called flipases. And you're like, nice, right? I got it. This one, it just scrambles them, like that. So, we're going to learn about 3, and that's 1. That event is happening. For that to happen, the scramblase is present, and it needs to be activated. And it's activated by caspases. And caspases are like the action in apoptosis. They're inside the cell, and if the cell activates caspases, it's dying. So that's the action. Those are the effectors of apoptosis. What we're going to talk about is upstream regulation of the caspases, because the caspases are in every one of our cells right now, every day of our life. Every cell is like ready to die. Yeah. It's got caspases, and they're like that. They're enzymes, and they're just like that, and they're being inhibited. right? Like that. And why do we have those? The interpretation is because if something goes wrong, you know, the cell starts growing out of control, get those caspases, activate them, flip the scramblase, you know, get it activated, flip it out like that. And we'll learn a couple other things, but, you know, we'll drop down to that molecular level and the regulation of apoptosis. Yes?
I just have a question about does every single cell have the phospholipid like every single cell? It's just that
Yeah,
apoptosis
in
is
every
like-
cell in our body, every day of our lives, every animal has that.
It's just that with apoptosis they stay in,
Yep.
stay
With
in
a healthy cell, they're on the inner leaflet, and when a cell goes through apoptosis, it's because the caspases get activated, and there are enzymes that activate things like scramblase, and scramblase flips it out. And then Godwin has our last question of the morning.
Are they just there for like a specific function or just to signal?
It's just that.
Just
Those are always a really good question, because it's surprising. Scramblase is there for one function, and it's there right now, and it's just inactive, but it's waiting. If that cell goes through apoptosis, it's going to get activated, and it flips it out, and that's it, right? And that's interesting to see, right? Because you're like, I know about hemoglobin that carries oxygen, you know, insulin, you know, right there, famous proteins, and they have a function. That's scramblase. It's just hanging out right now, just hanging out, waiting, inactive, and the cell is going to go through apoptosis. It gets activated, and of course, other things get activated. They're just waiting. So it's remarkable, but it's part of the evolution of multicellular organisms, right? We want to build this thing, but we want to build it in controlled ways. And if we get a cell that's out of control, doing the wrong thing, it should be able to kill, you know, itself. Upstream of caspases is regulation. It could be within the cell, like DNA damage, I'm going to kill myself, or it could be outside the cell, like a signal, like a hormone. Yep. So we got to go, but yeah, what a party! This is just a party, such a party, you guys.